Hi, everybody. So welcome to the podcast, Personal Genomics Zone. I'm very happy to have today Sean O'Donoghue, how I pronounce it properly. Um,
Perfecto.
so my first recollection of Sean was when I was a PhD student, actually. I was at Manchester University, and you were working for Lion Bioscience, a defunct company. And I remember that you had like already some kind of special hardware where you could visualize protein structures in a way that was very sort of innovative. So how long was that?
That was
20
20,
years for at least
25, maybe, almost even 25 maybe, because
25
2000,
years ago.
maybe 20, maybe 2001, yeah, something like that.
Okay, so Sean, tell me a little bit about yourself. So how did you get to what you are doing now? Tell me a little bit about your career and really help us understand where you're coming from.
Yeah. Well, Lion and that time that when I got to know you, that was actually the pivot of my career because before then I was like a mathematician, physicist, but I loved biology. So I was one of the classic bioinformaticians who come from the computation kind of side of things. And I was doing algorithms, but then I joined that company Lion and the human genome came out and we were working on that. It was really, really exciting.
Mm.
But I think I've got to press accept recording. I think hopefully it does record. um, hopefully we've got the first part. And then what happened was at Lion, I began to really think about, it's not enough if we can have the best algorithms you want and we can do the
Mm.
best analysis you want, but unless somebody actually sees the analysis, understands it,
Yeah,
then it's like the analysis didn't happen. And
right.
and then I realized it's also how we show scientists the data. So if we could do great analysis, but we've got to present the analysis results in a very clear way.
Hmm.
And it's never a single view, right? It's always got
Hmm.
to be interactive. So you're talking about interactivity in the software, and so it touches on design, it touches on the data visualization for sure, human visual perception.
Hmm.
And so since that time, really for me, it's a 25-year journey where I realized with a shock that the algorithms weren't enough. That we actually have to focus doubly hard because we've got to now create visualizations of the analysis results and interactions with them that are actually meaningful, accessible, and so it's a hard problem actually. It's a really hard problem, and but it's one that I just sort of I thought I was just excited by it because I think it's really fundamental to science moving forward to make sure that our data and our analyses are understandable. And so I've been on this bandwagon of trying to do what we can in the bioinformatics community to improve that. And yeah, that's very much what has brought me here today.
Okay. And tell me, in terms of the stuff that you've been able to discover as a consequence of applying these new methods, um, what have you been able to learn about biology in general and about science as well?
Oh, I mean, the first word that comes to mind is complexity. It's just
Mm.
because this, as we know in biology, the closer you look, the more you see.
Yeah.
And I think it's not only true just if you look at the sort of the physical, and in fact in data viz they've got a term for it. They talk spatial visualization. So you have a tissue, you look at that, and then you zoom down to the
Mm.
cellular level, and then you zoom down to the molecular level. But it's all spatial, so everything's just laid out there. And then of course
Mm.
you've got temporal arrangements and different slides, but fundamentally everything's just organized in space. But most of the data we do is non-spatial. It's like this abstract information about like genome organization or often we try to sort of merge the non-spatial to the spatial data, and there's so much complexity. That's the real thing I've learned. And I think most people don't really realize how much complexity there is, how much of it is unknown and undiscovered. And that's really interesting because I've also been involved in producing animations
Mhm.
where we try to explain what we know.
Mhm.
And the really interesting thing about when you make animations is you kind of have to fill in the gaps because you
Mhm.
show what you know, but there's all these things that we know we don't know. But if you're going to actually create an animation, you have to show something, and so you make your best guess, and that's really insightful. And when we make these animations, we work with world leaders in that field, like we've done them in Alzheimer's and various other, issues related to diet and so forth. And when we come back and we work with these experts very closely for months, and when we finally produce animation, the experts themselves are always surprised, and they learn something because they've never seen it before, even though they've been working in this for 30 years. Like we had this guy working on the human gut microbiome, and we asked him how many bacteria fit around a starch granule. So we have these granules of starch that come through into our large intestine, and we said, well, how many bacteria? Because we had to actually make the model and put a certain number. And he said, oh, he'd never thought of that question, like, because he maybe knows something about some overall stoichiometry, but had never actually asked physically how many would actually fit on one granule. And when you make these animations, when you try to really show it, you kind of have to fill in these gaps. And what, the big lesson I've learned is how much there is still to know. It's a shocking amount that we don't know.
One of the things that it has sort of struck me from having a conversation with you is how passionate you've been about all our previous exchanges as we were discussing this this presentation. I find that incredibly refreshing and, to be honest, enjoyable. So what makes you passionate about this? What's the, I want to understand what's behind, the real Sean O'Donoghue. I want to know what's made you so driven to be able to dedicate a whole life to these complex problems, and especially for people who might want to become scientists. I mean, being a scientist, rarely you're ever gonna get rich. Some of them do, but it's a different type
Very
of
rare.
life.
Yes.
It's a contemplative life, perhaps.
Yeah.
So tell me what's the motivation behind you being so passionate about what you do?
Well, I think for me, I think fundamentally, I'm excited by scientific mystery. If I see a mystery, and there's that, like, I've forgotten the exact languaging of this, is that wonderful thing of, like, is this actually, here's something we don't know. Is this actually impossible? Is this like beyond human comprehension or our ability to measure or even to calculate? Or is it something that just, if someone looks hard enough, they will actually discover something? And that excites me, just that idea that there's things to discover. And the more you realize, because that certainly has been my career, the more you talk to the people that are the most further along in their career, and I actually wanted to do this. I want to create a meeting. I have an idea a meeting to get the senior scientists and say, each of them to talk about the biggest mystery that is still out there. And I found many scientists have this. Many senior scientists have like one data set. I've seen this multiple times, and for many great scientists, and they show them one slide, say, we did this like 30 years ago, and we still have got no idea what we saw there. This result is remarkable, and we don't understand it. And I thought about, wouldn't it be great to have a meeting about these great mysteries? But yeah, the mysteries excite me personally. And then when you also realize that maybe I can do something, that's when it clicked for me 25 years ago that because I had this interest and in the technology of interactive visualization. I love that whole computing side. I love the programming side, and the idea that I could actually make an impact into one of these mysteries, or create tools that others could use to make an impact. It feels like the mystery is the drive, and then building the tools or building the solutions is just it just.
So how long have you been doing science, and what was the first thing that really hooked you when you decided to do a PhD?
Well, I was hooked long before then. I mean, I was always fascinated by life. I grew up in Australia. We lived on the sort of the edge of the towns. I was actually literally living in the desert as a kid, and I would go out into the desert and I would look at all the little things that are wandering around, the crazy wildlife we have in Australia. And the closer you look, the more you see. And I was gobsmacked at the what's going on in life, and I just wanted to know more. So that was, I always wanted to be a scientist. I think for me, there was sort of, again, a click moment where I realized that rather than studying biology as a biologist, and I found that a little bit, it didn't quite click with my brain chemistry or my brain organization. I'm more the mathematics, physics, programmer kind of person. But I realized that, hey, I could really apply all of that and help unlock some of these mysteries. And that was again where I realized I love the mystery, and I could do something about it. And I think once I found that click, for me, it was computational biology, bioinformatics. That was before the term bioinformatics really existed. I was hooked. And then about 20 years after that, I realized visualization is this big thing that brings everything together in one. It's kind of a symphony of everything. And that got me really excited. So here we are.
And tell me, so obviously this is going to be a topical question, but how things have changed since then in terms of how we approach science, and are we still asking the same questions?
To some extent, yes. I mean, let's go back to when we first met, 2001, 2000. The human genome came out. As mentioned, Lion was actually one of the companies involved in that analysis, and that was really exciting. And there was this idea back in 25 years ago, saying, okay, the genome's come out, and therefore that's it. Within a few months, we'll have sorted out all diseases. We'll have cured cancer, right? Obviously, that didn't happen, but because it unveiled this next, it's like a next level of the computer game, where we have this massive ton of information, and now we can actually see what the genome really is and begin to understand its complexity. And it's not obviously not going to be a rapid, necessarily rapid understanding. It's quite complex. Obviously, we have made huge progress. Like in medicine, most of my career has been involved in medical applications. I'm more on the science side, but I've always been close to medical applications. And cancer therapies, just many therapies have really marched forward, not in the pure cure sense, but the survival rates for all cancers have improved, and that's obviously and many other things have come from that moment of having the genome as a sort of a template, as a basis for organizing information has been really, really helpful. And then obviously all these other kinds of technologies that have come on board, the spatial transcriptomics, all kinds of omics, where we've been able to scale up the experiments and then so get good data at scale. And then the challenge becomes how to organize those data, and how to, again, the real question is, how do we put them together in a way that can enable the scientific community to find the data they need to answer specific questions. And that's what kind of motivates me. But yeah, so obviously we've got massive data. We've made huge advances in analyzing, understanding the data. But in many ways, the problem is bigger. Technology has improved. I mean, I personally love web developments, technologies. Web development stacks are amazing now compared with 25 years ago. It's computer power as well, but with the web in particular, it's moving like it's changing like gangbusters. It's a really interesting. I think you've been involved in some of those changes as well, right? You've been dabbling a lot in the web, I remember.
So I'm going to put this analogy to you. So we've been for a very, very long time, trying to have like translators, right? And we never quite got it right. I remember in the 2000s, you were trying to translate something from English into Spanish or something like that, and it wasn't really very good. And then Google, started doing statistics, but then, in 2022, we had the ChatGPT moment.
Yeah,
somehow we've cracked the natural language. We've cracked it with sheer computational power and a specific model that suddenly is able to speak better than most humans, at least in text.
Yeah.
And voice is pretty much like that. Do you think that we'll have a ChatGPT moment for life sciences?
That's an interesting question. Yeah. Well, I think I have to say that floored me too, because I always thought the language, I thought it was too complex. I kind of laughed at those guys. I didn't think they would get it. I thought they were deluding themselves, and I was wrong. Boy, was I wrong. And I would also say not just natural languages, but computer languages, and that's what I'm doing now. I'm doing prompting AI systems to generate code to create visualizations, and having a lot of success with that. Because I think potentially computer languages are maybe even better, more amenable to this because they are more logical, more structured. But yeah, I agree. If there's something has been unearthed, and I would not underestimate the data. It wasn't just the computational power, but it's the fact that there's just so much language, a corpus of language out there to draw on. And yeah, in theory, right? Because biology is a language, genomics is a kind of a language. In theory, that same answers, that same logic applies. That there's the corpora, which is the set of everything that's been sequenced, or everything that has existed that we can get our hands on, is out there. Perhaps where the analogy breaks down is because a lot of people probably have, I'm sure you know this idea, the idea that the genome is, it's not actually not a language. Somebody once described to me, this is the best analogy I have, which is the genome is actually, it's a compressed file. So you've got the real instantiated organism that's going out there, and then this is like the file of everything, but then this is highly compressed. And so it's not a language as such. We have to unravel the complexity of the compression process, because things don't, like in a truly true compression, things don't fit together that logically should. And so that's a complicating factor. Possibly it's the protein level, or maybe the RNA level, or even the protein, the post-translational modification layer. We've got to sort of work at reaching the right layer. And as someone who's my center of gravity has always been in proteins, I suspect the protein layer might be the right layer in which to really, where the real encoding is more like a language. Because when you look at systems of classic systems biology, language really doesn't begin to apply there. But if you look on the gene level, people have been struggling, and I've always been shocked as a protein person how bad predictions are on the gene level, compared with how accurate many protein, like a protein structure, can predict so accurately now. But the role of based on a pure genomic sequence, the role of the elements is still pretty poor by comparison with protein prediction.
So tell me a little bit then, since you are a protein guy, what has AlphaFold meant for you as a protein bioinformatician, and where is now the next frontier? Is Demis Hassabis going to take over all of the main problems, and we're gonna end up without no jobs? Or but where is the next thing now?
So I have a nuanced view, because this really comes close to home. My career has been evolving really close to the protein structure predictions. I know many of the players. I know the field, know how it's evolved. One of the key things I always want to remind people, before even AlphaFold came out, I've been singing the praises of the PDB, the Protein Data Bank, for many years. I've worked outside of the life sciences world, and I have never seen a database as well organized, as well curated. The idea that you have a single global collection of everything that's been published, and again, people complain that we don't have some of the things that have been done in companies, but there's no comparison to any other kind of data. I mean, I looked briefly with chemical databases. Everything is so fragmented. Systems biology, you've got 800. Last time I counted, 800 different databases on systems biology, and none of them really have any good way. Well, it's very hard to map between. With the protein structure database, it's extraordinarily well curated, and then the prediction methods, long before AlphaFold, we were running those prediction experiments, the CASP experiments, to really quantify the metrics of what is success. And then CASP came in on top of this incredibly well organized data, and even all these metrics that have been evolved over decades. And yes, they took a huge step, and it's exciting for the field for sure. But I think it's going to be hard to replicate that level of success for other fields, because simply their data is not as well organized. I mean, there's that old idea of garbage in, garbage out. And to some extent, what we were talking about before with the language models, this was surprising because I think what happened with language models is that they started to learn some principles beyond one language. I was recently testing ChatGPT with someone from the Basque country. There's only apparently about 500,000 native speakers, and we were testing its pronunciation and ability to understand his accents, and it was doing very well. And that was a surprise given how few, how there can't be that much literature out there, right? So yeah, I think that they managed to extract some general principles. But I don't know how that's going to extrapolate when we look at biology, because the different realms, I don't know if they're going to be as well interconnected. I'm still in awe of the complexity of biology, and but one of the best things I ever did was I met Frederick Brooks, who wrote the book Mythic Man Month. He's a very famous computer scientist, unfortunately recently passed. But we got to meet him a couple of times, and he had this beautiful statement. He's really said, if you had to predict, if you had an expert working together with an expert system, and they were competing against an expert system, a computer system by itself, which one would you bet on?
Mm-hmm.
Clearly, an expert together with an expert tool, an expert system, is going to, in general, outperform an expert system by itself without the guidance of an expert, a human expert. And I think that's the way to bet, that the AI systems they're really, really powerful tools, and they're going to help experts get more effective. But yeah, I don't know. Your question, I can't quite answer it. What's going to be the ChatGPT moment for the life sciences? I'm not sure.
Is it maybe that because ChatGPT happened because we had all of this available information on the internet, Wikipedia,
Yes.
Reddit, and we simply don't have that in biology, do we?
Yeah. Well, arguably we do, right? If we sequence everything and then determine the structure of everything, and then determine the transcriptomics profiles of everything, we can gather lots of information in theory, and we are getting good at doing that. There are problems. As I said, the data are not usually as well organized as the structural data. Even just the DNA sequencing data is not anywhere near as organized as protein structure data, which is surprising, actually. In theory, it could be much better organized. It's actually simpler data. But yeah, so I think I would not underestimate the role of the primacy, the impact of getting the data really well organized, really well structured, and that's not a technical thing. A lot of what happened in the PD was actually social. It was groups of people getting together who had that passion, who had that vision decades ago, and really working on formats, on conventions of how data is shared, so that they work together with the publishers to say, look, if we're going to publish a data set, you should give us a unique identifier, and it should be in the database. I mean,
so what's the next task for us who really care about understanding biology in a deeper level, now that we have this AI revolution? I've seen on the internet, like on Twitter, for instance, that some people were saying, the level of improvement of our understanding of biology has not sort of kept up with our developments in AI, and probably because AI is run by engineers who are cleverer than biologists. So I mean, what's your take on that?
Yeah, well, I think it's not fair. I mean, look, biology is intrinsically what's more complicated than life. Life is complex, and I've been talking about it before. So, like, let's talk about post-translational modifications of proteins or of RNA, or indeed of DNA. We know that we don't know. That the experiments have never been done in most cases. We cannot systematically measure most post-translational modifications across a whole proteome or RNAome or whatever. We can't do it. We have never done it. All of biology has been done without measuring the PTM state of proteins.
Can you explain what a post-translational modification is, just in case for those who might not know?
Right. Well, I like to think of them as switches. So a protein is made, but that's not the final story. It gets tweaked and modified, and sometimes drastically it gets cut into pieces. But very often there's these small little changes. They call them post-translational modifications, and often they're just like a little thing. And sometimes they're quite reversible. One, it's like a switch. It's like you have a complex, like a mixer in in a band or something, and you can switch things, turn off and on small parts. And proteins have got, I think, a million of at least 30 different. I think some proteins have hundreds and hundreds of different switches on one protein. And we haven't, all of biology has been done without measuring the state of proteins, because we can't measure those states at high throughput except maybe one kind of switch. And we know there's hundreds of switches, and we know and so we don't have the data. And so how can you have an AI system that's going to do something when the data isn't there to even analyze? And so there's a lot of innovation needed purely on the experimental side. Proteomics is really exciting, but then I've heard the RNA people saying there's even more post-RNA or similar kinds of modifications that happen in RNA.
And would you say, I mean, because the data in Wikipedia and the data in Reddit, which has one of the two main sources that have trained large language models, it's quite messy and it's quite unstructured. And if we were to use things like the UK Biobank, right, that contains, according to my last count, around 14,000 different types of data for 500,000 individuals.
Yeah.
If we were able to somehow handle all of that into a way in which we could feed a large language model, do you think that that would help us? Do you think that's possible? Would that help us get closer? If you had all of the computational capacity to process that, what type of visualizations would you work on?
Well, I think, I mean, the big question with machine learning in general is, are you using the right kind of machine learning? Like for a lot of the work that I've done, I've been using HMMs, not just as a user, hidden Markov models, not LLM, not large language models. And
so
what's
a HMM? What's a HMM?
Hidden Markov model.
Yeah.
Well, conceptually, I suppose, the overall architecture is if you, at a high level approximation is fairly similar, but it's based on this you take a sequence, and again, I've always seen it as very analogous to language, like a sentence. You have a sequence. You look at a protein as a sequence, or a piece of DNA, a gene as a sequence, and you try to predict what's going to come next. And you learn based on what happened earlier, what was observed earlier, what's the likelihood that you're going to see the pattern next. And by running through multiple families of related sequences, you then build up a sort of a sequence profile that gets encoded in those probabilities of the residue being next. And then it doesn't sound that overall, it sounds similar to large language models, but it's but the details of how these different encodings work are critical. And I don't know if I have, like you have, been surprised at the success of the large language models, but that doesn't necessarily mean that they're going to be the best model for approaching data, for example, about proteins or medicine and people's health records, where a lot of that is not actually encoded in language initially. I mean, health records to some extent, because to some extent they're actually written in English, but there is this question: to what extent is biological sequence information or other kinds of medical information is a language model the appropriate way? I suspect not. I suspect it's just more complicated, and it's going to be. Clearly, it's an advance. It's a huge advance. We can synthesize information we couldn't do before. But there's also the concern of this feedback cycle of now things become inferred from AI, but they're not necessarily determined from an experiment, and then that we don't necessarily cleanly annotate that distinction, and then we build new models based on not good solid experimental data, but inferred data. And we may be carrying across our errors in the future. So this is obviously a concern. And so visualizing the errors, I mean, I think that's a huge thing. That's one of the things. So the meeting I'm running We always try to have, we've had several speakers talk about the visual encoding of uncertainty and of errors, because it's one of those areas we all know we should be doing it more. And there's a whole group of community out there of visualization experts who are trying to develop good ways of encoding errors, and they've developed some fantastic stuff, actually. You should see this talk from Jessica Holman from our meeting last year. I found it gobsmacking how much progress has been made, but it doesn't translate across to the life sciences. Most of them are not using those techniques, and that's what we're trying to do.
Tell us more about your meeting. I remember it was with Niels Gellenborg, right? You
Yes.
organized it first,
Yes.
and then it's evolved. So tell us more about VizBi, the meeting that you basically, I would say, has defined a lot of what people know about you and what you are doing. So how did it start? And it's going to happen very soon, right? Tell us more about it.
Yeah, next week. Well, well, the origin was, so I have a partner in crime for everything with VizBi. It's Jim Proctor.
Yes.
What's his official title is essentially the lead developer on GalView, which is one of the world's most cited scientific papers, which is amazing. So it's a tool to look at multiple sequence alignments. And so he was also, like me, aware of the complexity of visualizing complex information and the difficulty of doing that right, and how we're often falling far behind I mean, if you look at Google, for example, or those that are really, Google Maps is regarded as one of the best data visualizations. It's an amazing story, the whole Google Maps. And what we need are tools in life sciences that have reached that level of utility. And that's a difficult thing because most of us don't have the resources of Google to develop that. And so we were at an ISMB meeting and realizing that we need to focus on visualization and in the life sciences. And it's mentioned, everyone's doing it, but people weren't really giving it prominence. And one thing we realized is that there's great data visualization researchers in computer science, and they never come to bioinformatics meetings. We never go to their meetings. And so either we try to sort of arrange an exchange of prisoners or something, but we came up with this model that would bring essentially those two communities together. And that's it. It's the 15th meeting next week. Every meeting's been really exciting because we're always bringing in people that have never been before. Every year we get fantastic. People love it because it's really is an eye opener just to see, if you're working in visualizing genomic information, to see what the protein people are doing, to see what tissue biology people are doing, to see what population people are doing. Because you're connected to them indirectly, but you can also learn the techniques and think, we could apply it to our domain. So we get that cross-pollination, and then to go and hear what the computer scientists are doing, these new techniques for visualizing uncertainty, for running all kinds of robust estimations with data and visualizing those outcomes, or the latest AI-driven data visualizations. What's going on there? It's and many people, if you see it visualized, it's easy to understand. So by forcing all of our speakers to focus on how they visualize their data, actually it becomes more easy to understand even outside of your field. And so it's a meeting. Not only can you learn about the visual aspects of it, we try to choose people from all areas of the life sciences who've got new data just in the last few years that have never been available before. And so they're really trying to work out what to do in terms of the analysis and also the visualization. The computer scientists come along because they love that idea of being involved in something new where they can contribute. And so it's a bit of a matchmaking service in some ways, connecting computer scientists. And then we had, and this really surprised me, we had the science communicators turn up, medical illustrators, animators, and they've made it their own. And it wasn't their idea at all at the beginning, but it's a very natural fit because they want to communicate the complexity of biology. They love biology and they love visual communication. And maybe they weren't that well organized. I don't know. They didn't have their own meeting, so they've sort of adopted our meeting as theirs. And I'm happy about that. It's a really nice fit. So it kind of rounds it out in a way.
So VizBi. So where is the next VizBi going to happen, and what are the highlights of this event
Yeah.
for this
We're
year?
very excited. Yeah, so for next week, we're very excited. We're coming to UK for the first time. We're in Cambridge University is hosting us at one of the colleges. Literally the town where molecular biology was born. I'm really, really excited about that. And we've got some.
City. It's not a town. It's a city.
Well,
It has a cathedral. It's a city.
well, I think it's arguable. I mean, maybe for me, it's like 1 million person is a sort of a cutoff, what I'd call a city. Just, I have memories of walking around 4 or 5 p.m. on a Sunday and not being able to get anything even to eat because everything was closed. And so that was,
I
as
love
someone from
Cambridge.
Sydney, I'm in love with this as well. Yeah, but I'm just saying from Sydney, where you have 5 million people, I don't quite consider Cambridge as on the same scale. So, but no,
No,
I'm
but
really
tell me
excited.
more about
Yeah.
this meeting. So it's going to be at
Yeah.
Cambridge University, which is one of the cradles of science,
Yeah.
literally. So that
Yeah.
in itself, you're surrounded by scientific discovery everywhere, and you're gonna bring some of the leading people doing visualization for biology. So tell us more. What do you have some kind of theme or thread for this event?
So an obvious thread is obviously the impact of AI on life science research, in particular how we're visualizing data, and that's definitely reflected in the keynotes. So that's and not just the keynotes. I mean, almost everyone, of course, is using AI for all the analysis. So that's a clear thread. But another one has just been it's one thing that's emerging in our community is the importance of I would call them visual metaphors. In fact, it's a paper I'm involved in working on at the moment on visual metaphors, because fundamentally we actually need to invent we have new data, totally new data, and some of it's abstract. It doesn't have a natural way in which we should express it, and so we've got to invent new ways of putting it down on paper, maybe interactively, and this is a human process. This requires creativity, and therefore we start to talk to the visual artists, and visual artists are engaged and interested from that point of view as well. And the beautiful example of this is from Jane Richardson, who was a speaker last year. She's actually coming this year as a normal attendee. We have amazing people like Jane Richardson, who invented the protein secondary structure representation, coming just as a normal participant. That's the amazing about VizBi. The community is amazing. But she invented that, and she invented it based on Greek pottery, and that was an art form that evolved, and then she saw this link in connection in how Greek motifs and so forth are expressed on pottery. It's so rounded around, a little bit like a protein, always kind of forms a globular shape. And so she invented this way of representing proteins. It's a human invention, but it's a really powerful one. And it's not directly related to the data. Again, this creative step was involved. And so this is where we need engagement of people that are really art focused. They want to see things in new ways. They're motivated to invent new metaphors. And this is definitely a strong theme this year. We've got 2 speakers who are multi award winners in things like Emmys, BAFTAs, and communications, and they're scientists who actually it was difficult to name them. We could maybe they're science artists, maybe, because like someone like Andy Lomez, he's not actually a scientist, but he really is a mathematician, but he loves science, and he's coming here. He wants to surround himself and learn, but you see that he's engaged in science, but he's not actually a scientist. So he's more of an artist, and so that is definitely a strong theme. And we have someone like Beata, if you haven't seen her work, so she's a cell biologist who wants to explain the weird and wonderful behaviors of cells by visualizing them in insightful ways. And she comes up with beautiful cartoons where you see things being teased apart, and proteins that cut things are shown as a pair of scissors, and it's a perfect combination of communicating the complexity of cell biology in a really beautiful way. that striking. You could just look at it as the artwork, but it's true science. It's true science communication.
So you're actually, it's not just about the visualization in itself. I bet that you guys get new perspective in terms of the biology, ways of understanding biology that is not necessarily just looking at graphs, right? So
oh yeah. Well, it's the continuum. It is the continuum. I mean, it starts with the bread and butter graphs, and there's many things we could do better there, and the uncertainty we were missing that, and it goes all the way up to the storytelling, communication, and then there's the conceptualization, because like the ribbon diagram of proteins is a way, because it's a way of seeing proteins that was invented by an artist, just effectively, and it affects how we now see and think about them. So the power of visual metaphors can be really striking. This is a really interesting conversation as well that we're having in the VizBi community over the last couple of years.
and is it still time to register?
Yes, it starts on Tuesday. We've got a 1 day. So basically, VizBi is a 3-day thing. We go through all of biology, and we do the central dogma. We start with DNA. We have a session on RNA visualization, proteins, cells, tissues, and ecosystems, and all kinds of populations. That's a 3-day thing, and that's a deep dive. But if you haven't got time for 3 days, many people don't. We've got this 1 day, what we call a masterclass, and that's where we have the 2 Emmy Award winners and some of the thought leaders in data visualization. So if you haven't got 3 days, just come and get 1 day. You can do it virtually. You can still attend in person. There's still a few seats available. We can sneak a few people in physically if you're close to Cambridge. But obviously, virtual registration is the most practical way for people. And yeah, virtual registration remains.
It'll be completely international, right? So what's the kind of breakdown? You're going to have a totally international audience, right?
Yeah, I mean, I haven't looked at the audience breakdown. The speaker breakdown is quite international. The VizBi attendance is always like that. Yeah,
how many people you get registered?
So typically, it's the smallest we've ever had is 130. The largest is 330, and okay. So 15 years, it's over just on 3,000 people, and so far have walked through the doors. And yeah, this year I think it's going to be it's definitely on the small side. This is a really tough year with all the cutbacks in the US, actually yes. The UK, actually France. Everyone it's tricky for people, but we've still got good numbers. I mean, they're low for us, but it's still going to be a great meeting.
So tell me a bit more about the current situation, political situation. Has it affected you? How do you see it from down there? And down under, I should say.
Well, I think my perception is very much skewed by, as one of the main VizBi people. I mean, and having run VizBi for 15 years now, and we're looking at the numbers and seeing that it's a tough time. People don't have the luxury of travel. Even just like in France, for example, we tried to do an event in France, but they've been cut actually totally independently of what's happened in the US, totally independent of UK. They've had cutbacks for the last several years that are huge cutbacks, billions of dollars of cuts to funds. And so one of the first things to cut is because people, rather than cutting entire people, you just reduce how much money everyone has. And so things like training, conferences and so forth, I think that's the first thing to cut back. But science really does depend on the exchange of ideas. And I think all of us, we've tried to do it remotely, and there's a lot you can do there, but it's tough. And I think we'll never really replace the in-person experience. The real question is, how do we optimally combine that with how do we, because not everyone can come in person, and so how do we give the people that can only attend virtually, how do we give them a taste of that in-person experience? But we also have to make the in-person experience still work. Otherwise, I mean, we could of course go fully virtual, but you really lose a lot then. So that's what I know is I look at the numbers, and I also talk to other media organizers, and everyone's seeing the same thing. And so it's a struggle to work out how to adapt with the current situation.
And how is yourself? How is Sean adapting to? Do you feel a little bit What's your feeling? What can we do as scientists to protect the things that we value so much? This
Yeah. Well, I think the only thing that, at least for me, the way my mind works is these situations. I think we have to look at what we have, what is working, because some things are still there, and many things actually work. Right? Actually, science is better than it's ever been. The internet's the movement of all literature online. I mean, this is even you. I'm quite a bit older than you, but even you are old enough to remember the time when most literature was actually not available online, and now, of course, that feeds into things like the large language models. So I think we have to count our blessings, really. And yeah, it's a time of change, and we're going to lose some things. So we have to fight for the things that we think are valuable for, fighting for, and try to weather the storm. And there will be some things we will lose some things along the way. I mean, some data resources are being closed.
what things are worth fighting for?
Well, I think everyone has their own priority list, and
What's your
like
priority
this.
list?
Well, of course, I would love to see us prioritizing how we visually organize our information. I think that's one of the big things we're not doing. I recognize that this is a tough priority at this stage. So hunkering down. I think in my own little world, the fact that we've made VizBi a success, that we're going to have 130 people next week. That's actually fantastic. It is a sign that we have a community here that does value it to turn up, either virtually or in person. So yeah, I think we have to, and maybe it's what's that saying? You don't realize the value of what you have until you risk losing it, and then you realize, okay, this is something I really want to keep. And so yeah, for me, it's VizBi. It's been a tough fight, to be honest, to keep this thing going. We fought harder than ever before, and but we know it's worth it. So we've done the experiment. This will be the 15th time, and as mentioned, when you get a group of people together in a room, many of whom haven't met but have this common goal, it can be tremendous what comes out. And maybe that's like an upbeat note to say is that human ingenuity. Let's not underestimate that. We're scientists. Amongst our community, there's some incredibly ingenious people. So if we put our minds together, we can come up with great solutions. And maybe we have to refocus some of our efforts from the pure endeavor of science to how do we fund science? How do we keep scientists engaged when we have all these cutbacks? There are ways. I myself am very excited about things like crowdsourcing, and I've been building in aspects to that to my work for a long time, and now I think it probably is the time to scale these kind of attempts up. A lot of science can actually be done by people that aren't necessarily working full time as scientists, and that's an interesting thought.
So usually when I close this type of meetings, I always want to ask this question, last question to all my speakers, and that question is, if you were to talk to your younger you when you started your PhD, what would you say to your younger self?
Oh boy!
What advice would you give to your younger self 30 years ago?
Wow, I mean, it's probably more like 40 years actually in my case. So, and there's been so much happening in the meantime in my own evolution, the things I've taken on. I wouldn't know where to start. I mean,
I like to make it a little bit philosophical because
okay.
I'm as much interested in the person as in the science, so I cannot differentiate between the two, and I think understanding the person behind makes science more attractive. Actually, I think we forget the social aspect of science, how important
Oh yes, yes.
The human connection and
Yeah,
relationships are
yeah. And
fundamental part of how we do science. It's not
yeah,
about the people in the lab just doing their own
yes,
thing.
yes. And I totally with you, and that gives me the framework for an answer. Because I think one of the things that I didn't do when I was young, I literally grew up in small towns, a totally non-academic background. I'm one of those people I just wanted to be a scientist and just had to sort of find my way there. I wasn't ambitious enough because I'd never been given a frame of reference to be ambitious, and I somehow stumbled into EMBL, where we had people playing at the top level of the game, and then in bioscience, we were literally doing some of the first analysis of the human genome. And ever since those experiences reset my goals. So let's take on a really big challenge. I think that's probably the big thing I would say. And part of taking on that big challenge is finding other people who have a similar mindset and somehow working with them, choosing them as a supervisor, somehow just getting in their orbit. And what I've done, part of my motivation to make these meetings work is to enable that bringing together of people so that kind of connection can happen. Because I think it's not going to happen by itself. It's about connecting people.
In order to do that, you need to think a little bit out of the box. And I have the feeling that you are one of those people who are not necessarily fitting the mold. Am I right, do you think?
I think so, yeah. Definitely. Well, I noticed this already in 2000 before line. When I went to bioinformatics meetings, everyone thought I was in ICMP. Everyone thought I was the biologist. So I mean, with computer people, they think I'm a biologist, but when I'm with biologists, they think I'm a computer guy. And now I've added this visualization layer to it, and I'm often between these worlds and seeing the value of both and trying to sort of connect them and trying to learn from both.
Sean, I
And
will never forget, I promise you, I will never forget that when I was a nobody, a student, you actually took me seriously. You showed me things, you explained to me things, you engaged with me when I was just starting. And I have not forgotten that. I promise you. I promise you that. I know
well,
it may have been a tiny thing, but
yeah,
you took me seriously. And there are things that you forget. I don't forget. I still remember the room. It had the hardware. It had like some kind of, I don't know, like so I still have it in my mind. So
yeah.
I think that the fact that you didn't see that student, you didn't see someone who's got this weird accent, you didn't see some, actually you you just came and talked to me as someone else about science, and you were excited to show me all of that. I think that is something that certainly I would say is very valuable that you have.
Well, thanks for giving it back to me, to reminding me of that, because so many things like that, they sort of get lost in time, and not many people take the time as you've done to really thank me, and that so that is actually very rewarding. And I should do the same and pay it forward, because there's so many people that have motivated me in the same way, and I should pay it forward more, because it's easy to get wrapped up in our own stuff and forget about the impact that we have on others. But yeah, that's a gift of enthusiasm, the excitement about someone who's passionate about their work, who loves it, and that's the thing. It's hard to break across the wire sometimes. Maybe we can do it to some extent, but that's why I'm passionate about these meetings as well, because it enables that kind of communication to happen. Well, thanks, Manny.
So I'm
Thanks for
so
reminding me.
glad. So I'm really happy to be able to catch up with you, have this friendly pub-like conversation. I'm sure there'll be more in the future. So I look forward to hearing from all the amazing outcomes that will come from VizBi 2025. Maybe if you want to tell a final message for people, if they want to join the conference, Visualizing Biological Data, it will be in Cambridge, Magdalene College, 8th to the 11th of April.
Yep.
you still can register. We'll put this this weekend, so there'll be a few days left. If you
Yep.
cannot come to Cambridge, you can do it online. Anything else you like to say?
Yeah, so via VizBi.org. So we've also got hundreds of videos because we've put a lot of work into making, unfortunately, not all of them are available and not all of them are free, but most of them are. So fantastic, amazing speakers just there. We've got also 1,000 posters over 15 years. So people have contributed, great ideas. It's getting so large, it's hard to load that page, but we're going to work on improving that. So there's a lot of resources there for the community. And we have actually, we're going to announce it. So if you go to VizBi.org, it's going to be advertised there. One of the talks is going to be freely streamed, and there's going to be several that will be made available freely as well. So this one is by Laura Garrison. So she's talking about visual communication and how we, as scientists, whenever we create visualization, what's that process we go through, and what's better ways of improving that process. That's literally what she studies, that exact process. And that's on Thursday next week, 6 p.m. UK time. It's going to be streamed live, and then the video will be available within 24 hours on the website, freely available. So if you can't afford the time or the money to even sign up for the masterclass, then at least you can catch that one.
Thank you. And I guess you have a pint at the Eagle, right? I mean, that's
always do. I have to do that.
You have to do that. Is it table 12 or table 16? The actual one. I can't remember the exact, but there's the table, and you can sit there. I actually sat there like a few months ago. I took a video, so that you still can go there. I'm sure they will close it at some point, like they do with all the good things. But
it's a bit magical for me because I went to visit a friend there who worked at EBI, and there was a big crowd outside, and I had difficulty getting in. There were so many people, and then just as I was about to walk in the Eagle, I turned around, and just behind me was Stephen Hawking.
Wow.
He was just opposite the Eagle. There's this amazing clock that you might know of, and it was the unveiling for that. And so I
never got the point of that clock. I mean, it's some kind of but it's like a what's the point of the clock? It doesn't even tell you what the time is. I cannot tell by looking at that clock. What's the story about that clock?
I thought the story was something about, because I have a friend who's actually works on creating pendulums that operate differently, and I thought there was something about the pendulum, but I guess I will go to the Eagle and I'll make a point of trying to find out not only the table one, but also about
let us know for the next time. Okay, Sean, thank you so much for talking to me, and I wish you all the success for VizBi 2025 and for the many years to come.
Thank you, Manny, and let's definitely catch up ourselves for a pint.
Yes, we will.
in real life. All right.